Absolutely. Here's the greatest, deepest, most detailed summary of the entire passage you gave, explained in the simplest, clearest way possible, like a world-class teacher teaching someone with zero background in science. Detailed summary of the passage in the simplest language. 1. What is biology? Biology is the science of life. It means studying all living things like humans, animals, plants, and even tiny microbes. It helps us understand what living things are made of, how they grow, how they stay alive, and how they behave. 2. Living things look very differently. When scientists studied living things, they saw a huge variety. A tree looks very different from a human. A fish is different from a bird. This variety is called diversity, meaning many different forms and appearances. 3. But they all have one thing in common: cells. Even though living things look different, they all share one common feature. All living things are made up of cells. This idea is called the cell theory. A cell is the basic unit of life, like a building block. Just like a wall is made of many bricks, a body is made of many cells. 4. What is a cell? A cell is a tiny living unit. You can't see it with your eyes. You need a microscope. Some organisms have just one cell, like bacteria, while others have trillions of cells, like humans. Each cell can take in food, grow, make energy, reproduce, do special jobs. 5. Living functions need a complete cell. Scientists found that life processes only happen in complete, healthy cells. If you break a cell into parts, the cell cannot do any living work, like breathing or thinking. Like a mobile phone. If you remove the battery or screen, it won't work, even if you have all the parts. 6. Studying life using physics and chemistry, physico-chemical approach. To deeply understand how life works, scientists used ideas from physics, energy, motion, light; chemistry, molecules, reactions. This is called the physico-chemical approach. They studied what's happening inside the cell, using pieces of the cell or even parts taken out of cells. This helps explain how cells work in terms of molecules and chemical actions. 7. Breaking life down to molecules. Using this method, scientists began to explain life using molecules. A molecule is a tiny chemical particle like sugar, protein, fat, or DNA. These molecules are the workers inside a cell. They make life happen. 8. Finding what molecules are in living things. Scientists took small pieces of living things called tissues and studied what chemicals they were made of. They found elements like carbon, hydrogen, oxygen, compounds like water, proteins, sugars. They identified organic compounds, which are the main molecules found in living organisms. And 9. Types of organic compounds in life. Biomolecules. These are the molecules found in all living cells. 1. Carbohydrates like sugars give quick energy. 2. Proteins help build body parts and do work inside the cell. 3. Fats, lipids, store energy and protect organs. 4. Nucleic acids, DNA and RNA, carry genetic information. Together, these are called biomolecules, the molecules of life. 10. What do these molecules do in a cell? After finding out what molecules are present, the next step is to ask, what do these molecules actually do? How do they help the body work? 11. Molecules help in big body functions. These tiny molecules inside cells do big and important jobs, like examples: digestion, breaking food into small parts; excretion, removing waste like urine, sweat; memory, storing thoughts in the brain; defense, fighting harmful germs; recognition, knowing which cells belong to your body and which are foreign. All these processes happen because of molecules inside your cells. 12. What is the molecular basis of life? Scientists now try to answer, how do molecules inside cells make life happen? This is called the molecular basis of life. It means explaining breathing, eating, healing, thinking, and everything else in terms of molecules doing their jobs. 13. Understanding diseases using molecules. When these molecules stop working properly, we get diseases. Examples: if the insulin molecule doesn't work, person gets diabetes. If cell growth molecules go out of control, So studying molecules helps us understand what goes wrong in a disease and how we can fix it with medicine. 14. This is called reductionist biology. This approach, breaking big living systems into small parts, is called reductionist biology. That means reduce the complex body into simpler parts, like molecules. Study each part deeply, then understand how the whole body works, like a mechanic who opens a machine and checks each part to understand how it works. 15. Using physics and chemistry to understand biology. To use reductionist biology, we need tools from physics, like microscopes, energy detectors, tools from chemistry, like chemical tests, reactions. Together, they help us understand life in the deepest possible way, from the smallest building blocks. 16. Learn more in Chapter 9, Biomolecules. The book gives a special chapter to help you understand biomolecules, the most important molecules of life. That chapter will teach what biomolecules are, what types exist, sugar, protein, fat, DNA, and what each one does in the body. Final deepest summary. In just a few sentences, biology is the science of life. Even though living things look different, diversity, they are all made of cells, unity. Cells are the smallest parts of life. Life only happens when the whole cell is working properly. To understand life, scientists use tools from physics and chemistry. They study the small molecules inside cells. These molecules, called biomolecules, do all the work like digestion, memory, and defense. If they don't work properly, we get diseases. This method of breaking life down into small parts to understand it is called reductionist biology. Let me know if you want a comic strip, diagram, mind map, or questions and answers to help you revise this better. Of course, here's a great, deep, detailed summary of everything you shared about G.N. Ramachandran in simple, clear language, but with full detail so you understand both his life and why his work is so important. Think of this like the best notes you could get from a top teacher.
Who was G.N. Ramachandran?
G.N. Ramachandran was one of India's most brilliant scientists. He worked in the field of protein structure, which means he studied the shapes of proteins, the building blocks of our body. His discoveries helped scientists all over the world understand how proteins fold, function, and stay strong.
What did he discover?
Triple helix of collagen. Collagen is one of the most common proteins in our bodies. It is found in skin, bones, cartilage, tendons, and teeth, giving strength and flexibility. Before Ramachandran, scientists didn't know exactly how collagen was shaped. In 1954, he discovered that collagen is made of 3 chains twisted together like a rope. This is called a triple helix. This discovery was published in the top science journal Nature and changed the world's understanding of how structural proteins work. Ramachandran plot. Proteins don't just stay straight, they fold into complex shapes. The way they fold is critical. If they fold wrong, they don't work or cause diseases. Ramachandran created the Ramachandran plot, a special chart that shows which angles of folding are allowed for a protein, which angles are impossible because they would cause parts of the protein to clash or break. His plot became a universal tool in biology, still used today by scientists worldwide to check and predict the shapes of proteins. The Ramachandran plot is so famous that every biology student learns about it. Founder of the Madras School of Conformational Analysis, Ramachandran started a research group in Madras, now Chennai, India. His group became world famous for studying biopolymers, long chains of molecules like proteins and DNA that make up living things. They specialized in conformational analysis. Conformation equals the way a molecule bends and twists. Analysis, carefully studying it. By analyzing these shapes, they helped understand how life works at the molecular level.
Early life and education.
G.N. Ramachandran was born on October 8, 1922, in a small town near Cochin, Kochi, on the southwestern coast of India in Kerala. His father was a college mathematics professor, which meant Ramachandran grew up in a home that valued education and logic. This inspired him to love mathematics from an early age, which later helped him calculate and describe protein shapes.
His academic journey.
He finished school and went to the University of Madras. In 1942, he graduated with a BSc honours degree in physics, standing first in his class, a sign of his brilliance. He then went to Cambridge University in England, one of the most famous universities in the world. In 1949, he earned his PhD, becoming an expert in physics and applying it to biology. Influences that shaped his career. While studying at Cambridge, he met Linus Pauling, a legendary American scientist who discovered the alpha helix and beta sheet structures in proteins, shapes critical for life. Pauling's groundbreaking work inspired Ramachandran to study collagen, leading to his own historic discovery of the triple helix. Why his work matters today. Proteins must fold correctly to work. If they fold wrongly, it can cause diseases like Alzheimer's or cystic fibrosis. The Ramachandran plot helps scientists check if protein structures are folded in a way that can actually exist in nature. Understanding the triple helix of collagen has helped medicine, dentistry, and cosmetic science, since collagen is used in many treatments and products. His death and legacy. Ramachandran died at the age of 78 on April 7, 2001, but his contributions still live on. His discoveries are taught in schools and used in labs around the world every day. He put India on the global map in the field of structural biology and inspired generations of scientists. Key takeaways. G.N. Ramachandran discovered that collagen has a triple helix shape, 3 chains twisted together like a rope. He created the Ramachandran plot, a chart showing which folds of proteins are possible and which are not. He founded the Madras School of Conformational Analysis, making India a centre for advanced study of biopolymer shapes. His work helped science understand how protein shapes relate to health and disease. He was influenced by great scientists like Linus Pauling, but he made his own historic contributions that changed biology forever. Why should you remember him? Without knowing the correct shapes of proteins, doctors and scientists couldn't design many modern medicines or understand genetic diseases. Ramachandran's work forms the foundation of structural biology, which connects chemistry, physics, and biology. He is an example of how passion, curiosity, and math skills can lead to world-changing discoveries, even if you come from a small town. That's your great, deep, detailed summary in the simplest language. Would you like a timeline, comic strip, or a quiz to help you remember all this? Of course, here's your great, deepest, detailed summary of everything you just shared, in clear, simple language, but with complete detail, like a set of perfect notes from the world's best teacher. Living versus non-living things. When you look around in your home, school, park, you see 2 types of things. 1, living things, like humans, animals, plants, insects, bacteria. 2, non-living things, like stones, chairs, water, books. This difference raises an important question. What makes something alive? Why can a dog eat, grow, and reproduce, but a rock cannot? The main difference. Cells. The main thing that makes something alive is the cell. A cell is the smallest basic building block of life, like a tiny living brick. Living things have cells. Non-living things do not have cells. Organisms. Unicellular and multicellular living things are made of cells, but there are 2 types. 1, unicellular organisms, made of only 1 cell. This single cell does everything: eating, moving, growing, reproducing. Examples are paramecium. 2, multicellular organisms, made of many cells working together. Cells divide the work. Some cells digest food, some help sense the environment, some help move. Examples: humans, dogs, trees, fish. Why cells are special? A whole cell can perform all life functions. It can get food, use energy, grow, reproduce, and respond to the environment. Anything less than a complete cell, like just a piece of its membrane or just the nucleus, cannot live independently. That's why the cell is called the fundamental structural and functional unit of life. Cells build the body of things like bricks make a house. Functional unit, cells do all the work needed to stay alive, like factories producing everything a body needs. History of discoveries. Antony van Leeuwenhoek, late 1600s, made one of the first simple microscopes. First person to see and describe living cells, he called them animalcules. Saw bacteria and protozoa in pond water. Robert Brown, early 1800s, discovered the nucleus inside plant cells. The nucleus is like the brain or control center of the cell. It holds DNA and controls all cell activities. Importance of microscopes. Without microscopes, humans cannot see cells. They're too small. First microscopes showed the basic shape of cells. Improved microscopes let scientists see more details. Parts like cell walls, cell membranes and organelles, tiny parts inside a cell. Electron microscopes, invented in the 1930s, can see objects millions of times smaller than the naked eye can see. Revealed even tinier parts like mitochondria, energy factories of cells, ribosomes, protein-making parts, and endoplasmic reticulum, transport system inside cells. Microscopes changed our understanding of life. They showed that every living thing is made of cells and helped scientists learn how cells work and what goes wrong in diseases. What cells do. Cells perform all the basic life processes, taking in nutrients, food, breaking down food to get energy, building new parts and repairing damage, getting rid of waste, reproducing to make more cells, responding to changes like moving toward food or away from danger. In multicellular organisms, cells become specialized. Muscle cells help movement. Nerve cells send signals. Blood cells carry oxygen.
Why cells are important?
Everything alive, from the tiniest bacterium to the tallest tree to the largest whale, is made of cells. Cells are the basic unit of life. Life cannot exist without them. If you break a living thing down, the smallest piece still alive is a single whole cell. Cells explain why living things can grow, heal, and reproduce, and why non-living things cannot. Deepest simple summary. 1. Living things have cells, non-living things don't. 2. The cell is the smallest part of a living thing that can do everything needed for life. Some living things are unicellular, one cell area. Others are many cells like humans. 4. Parts of a cell cannot live alone. Only a complete cell can. Scientists like Leeuwenhoek and Robert Brown helped us discover cells and their parts. 6. Microscopes were essential for seeing cells and learning how they work. 7. Cells are called the fundamental structural and functional units of life because things and do all the work to keep them alive. That's your great, deepest, detailed and simplest summary of what makes something alive. The cell? Would you like me to create flashcards, a mind map or quiz questions to help you remember this? Of course, here's your great, deepest, most detailed summary of the whole section you shared on cell theory, explained clearly but thoroughly, like perfect notes you can review anytime. This summary brings together all the points in one place so you never miss a detail. In the 1800s, scientists began to study living things with microscopes and realized life is made of tiny units called cells. Matthias Schleiden, 1838, German botanist, plant scientist. What did he do? Examined many plants under a microscope. What did he find? All plants, no matter how big or small, are made up of cells. Key idea: cells form the tissues of plants, like leaves, stems, roots. Impact: first big step showing plants are built from cells. Life has a cellular basis. Theodore 39.
Who?
German zoologist, animal scientist. What did he do? Studied different types of animal cells. What did he find? All animal cells have a thin outer layer, which he identified as the plasma membrane, the cell's protective skin. By comparing with plant cells, he saw plants have an extra thick layer outside the plasma membrane, the cell wall, a unique feature of plant cells. Key conclusion: both animals and plants are made of cells, but plant cells also have a cell wall. Impact: first scientist to clearly connect animals and plants by saying both are made of cells. Schleiden-Schwann formulate cell theory. Together, Schleiden and Schwann combined their findings. They proposed the first version of cell theory, which said, 1. All living things, plants and animals, are made of cells. 2. These cells are the basic building blocks of life. Impact. This was revolutionary. It unites study of plants and animals under one fundamental idea. The cell is the basic unit of life. Missing piece: how are new cells formed? Although Schleiden and Schwann showed all living things are made of cells, they didn't explain where new cells come from. At the time, many believed in spontaneous generation, the false idea that living things could suddenly appear from non-living matter, like maggots appearing from rotting meat. Who? German physician and pathologist. What did he do? Solve cell theory. What did he prove? New cells come from the division of existing cells. Cell not a famous phrase, omnis cellula cellula, Latin, meaning every cell comes from another cell. This completed cell theory and explained how growth, healing, and reproduction happen. The final modern cell theory as we understand it today includes 2 key points. 1. All living organisms, from bacteria to humans to plants, are made up of cells and the things cells produce, like fibers, enzymes, hormones. 2. All cells arise from pre-existing cells by the process of cell division. New cells come only by splitting from existing ones, not by appearing magically. Importance of cell theory. Proves that cells are the smallest unit of life. They do everything needed for living. Explains growth. Plants and animals grow because cells divide to make more cells. Explains healing. When you get a cut, new skin cells come from dividing existing cells to repair the wound. Helps doctors understand diseases, since many health problems start with cells dividing too fast, like cancer, or not dividing properly. Shows that all living things share a common feature: cells, the universal building blocks of life. Complete, deepest, simple summary. In 1838, Schleiden discovered all plants are made of cells. In 1839, Schwann discovered all animals are made of cells and identified the plasma membrane. He also noted plant cells have a cell wall, unlike animal cells. Together, they proposed the first cell theory, stating all living things are made of cells, but they didn't know how new cells formed. In 1855, Virchow completed the theory by proving new cells come from existing cells, with his phrase, omnis cellula e cellula. The modern cell theory says, 1. All living things are made of cells and what cells produce. 2. New cells come from existing cells through division. Why you should remember this. The cell theory is one of the most important ideas in biology. It explains how life grows, heals, reproduces, and evolves. It unites all living things, from tiny bacteria to giant whales, under the same principle. Everything alive is made of cells. Would you like a timeline chart, a comic strip, or some flashcards to help you remember this easily? Of course, here's your great, deep, detailed summary of the whole explanation you shared, laid out clearly, but with full depth so you have every important point in one place. The experiments. You've probably seen onion cells from a thin peel and human cheek cells scraped gently from inside the mouth under a microscope. These experiments show what plant cells and animal cells look like in real life. Onion cells, plant cells, onion cells are plant cells which have cell wall, thick, rigid outer layer, made of cellulose, provides shape, strength, and protection. Cell membrane, thin, flexible layer just inside the cell wall, controls what enters and leaves the cell. This double boundary, wall plus membrane, gives plant cells their box-like shape. Cheek cells, animal cells. cheek cells, which have only a cell membrane as the outermost layer. No cell wall. Animal cells are more flexible and can have irregular shapes. The nucleus. Inside both onion and cheek cells is the nucleus, a dense membrane-bound structure. The nucleus is usually the most noticeable part of the cell, has its own nuclear membrane separating it from the cytoplasm, contains chromosomes, which are made of DNA or carrying genetic information, controls the cell's activities like a manager's office. DNA and chromosomes. DNA in the chromosomes holds genes, instructions that determine traits and direct how the cell works. DNA looks like a twisted ladder, double helix, and is the same in all living things. DNA, chromosomes, nucleus, cell, whole organism. Types of cells. Eukaryotic vs prokaryotic. Eukaryotic cells have a true nucleus surrounded by a membrane, store DNA inside the nucleus. Are complex cells. Examples include plants, animals, fungi, and protists. Prokaryotic cells do not have a nucleus. DNA floats freely in the cytoplasm. Are simple cells. Examples include bacteria and archaea. This distinction is fundamental. Eukaryotes equal cells with nucleus. Prokaryotes cells without nucleus.
The cytoplasm. Cytoplasm.
Semi-fluid jelly-like substance filling the space inside the cell membrane. Occupies most of the cell's volume. Holds all organelles in eukaryotes or cell components in prokaryotes. Cytoplasm is where most of the chemical reactions needed for life happen. Cellular reactions in cytoplasm. The cytoplasm is the main arena of cellular activity, breaks down food for energy, builds new cell parts, processes waste products. Without reactions in the cytoplasm, cells couldn't stay alive. Organelles in eukaryotic cells. Eukaryotic cells have specialized organelles, each with a specific job. Endoplasmic reticulum, ER, network of tubes, makes and transports proteins and fats. Golgi complex sorts, modifies and packages proteins and lipids. Lysosomes contain digestive enzymes to break down waste or damaged cell parts. Mitochondria produce energy by breaking down sugars, called the powerhouse of the cell. Vacuoles store water, nutrients, or waste, larger and more prominent in plant cells. Microbodies, small vesicles with enzymes for various reactions, e.g. peroxisomes for breaking down toxins. Prokaryotic cells lack organelles. Prokaryotic cells do not have membrane-bound organelles. Everything, DNA, proteins, enzymes, is mixed together in the cytoplasm. Their processes happen directly in this open jelly-like environment. Complete summary in simple points. 1. Onion cells, plant cells, have cell wall plus cell membrane. 2. Cheek cells, animal cells, have only a cell membrane. Both have a nucleus with chromosomes made of DNA which carries genetic instructions. 4. Eukaryotic cells have membrane-bound nuclei and organelles, plants, animals, fungi. 5. Prokaryotic cells lack membrane-bound nucleoles, bacteria. 6. Cytoplasm is the jelly filling the cell where life's reactions occur. Eukaryotes have organelles like ER, Golgi, lysosomes, mitochondria, vacuoles, and microbodies. Prokaryotes don't have these organelles, they're simpler in structure.
Why this matters?
Understanding these basics helps explain how cells grow, heal, make energy, and pass on traits. These differences are why bacteria behave differently from plants or animals. It's the foundation of biology. Cells are the smallest units of life, and their structures determine how life works. Would you like a mind map, comic strip, or flashcards to help you memorize this easily? Of course, here's your great, deepest, and most detailed summary of everything in the passage, but still in simple, clear words so you get the full meaning without confusion. Ribosomes. The universal protein factories. Ribosomes are tiny round structures inside every living cell. They do not have membranes around them. They're bare organelles. They are found in all living cells. Prokaryotic cells, bacteria and archaea. Eukaryotic cells, plants, animals, fungi, and protists. Ribosomes make proteins, which are essential for life, like bricks, machines, and tools for the cell. Where ribosomes are located. floating freely in the cytoplasm, where they make proteins used inside the cell. Stuck on the rough endoplasmic reticulum, rough ER, where they make proteins sent out of the cell or to the cell membrane. Inside mitochondria, in all eukaryotic cells, and chloroplasts, in plant cells, where they make proteins for those organelles' own needs. Centrosome, the cell division organizer. The centrosome is another important non-membrane organelle, but it's found only in animal cells. The centrosome includes two tiny tubes called centrioles sitting at right angles to each other. During cell division, the centrosome organizes and controls the formation of spindle fibers. These fibers pull chromosomes apart, so each new cell gets the correct DNA. Cells come in many sizes. Cells show incredible variety in size, including extremes: smallest living cells, mycoplasmas, only 0.3 micrometers long, so small they're near the size of large viruses. Typical bacteria, 3 to 5 micrometer long, larger than mycoplasmas but still microscopic. Largest single cell in nature, the ostrich egg, a single cell you can see and hold in your hand. In multicellular organisms, human red blood cells, RBCs, are about 7 millimeters in diameter, small enough to move easily through tiny capillaries. Human nerve cells, neurons, can be over 1 meter long, stretching from your spinal cord to your toes. Cells have many shapes, each for a purpose. Cells aren't all round or square. They come in many shapes because the shape matches the cell's job. Examples include disc-like cells, like red blood cells, thin and round so they can squeeze through the smallest blood vessels. Polygonal cells, like skin cells, many-sided shapes that fit tightly together like tiles to protect your body. Columnar cells, like cells in your intestine, tall and thin, ideal for absorbing food nutrients. Cuboid cells, cube-like shapes, seen in kidney tubules, good for absorbing and filtering fluids. Thread-like cells, like muscle fibers or nerve cells, long and thin, perfect for movement or sending signals. Irregular cells, like white blood cells, can change shape, which helps them move and chase bacteria. Shape matches function. A cell's shape is not random. It's perfectly designed for its specific job. Flexible disc-shaped red blood cells carry oxygen efficiently. Long nerve cells allow fast communication across the body. Shape-changing white blood cells can move through tissues and attack germs. Complete, simple, deep summary. Ribosomes are small non-membrane organelles in every cell, making proteins needed for life. Ribosomes are found free in the cytoplasm, attached to rough ER, and inside mitochondria and chloroplasts. Centrosomes are non-membrane organelles found only in animal cells. They organize cell division by controlling spindle fibers. Cells can range in size from the tiny mycoplasmas, 0.3 micrometers, to the huge ostrich egg, the largest single cell you can see. Human red blood cells are about 7 micrometers wide. Some nerve cells are over 1 meter long. Cells come in many shapes: discs, columns, cubes, threads, polygons, and irregular forms, each matching the specific function of the cell. A cell's size and shape are perfectly adapted to its job, allowing living things to have diverse, specialized cells working together. This summary gives you every key point with added clarity and examples so you fully understand how ribosomes, centrosomes, cell size, and cell shape connect to cell function. Would you like a visual diagram, comic strip, or flashcards to help memorize this? Of course, here's a great, deepest and fully detailed summary of everything you just read about prokaryotic cells, but still in simple, clear words with every key point included so you can understand it completely. What are prokaryotic cells? Prokaryotic cells are the simplest and most ancient type of living cells on Earth. They do not have a true nucleus. Their DNA floats directly in the cell's jelly-like inside, called the cytoplasm. Prokaryotic cells are found in bacteria, tiny single-celled organisms everywhere in nature. blue-green algae, cyanobacteria, bacteria that can do photosynthesis like plants, mycoplasma, the smallest living cells known, PPL, pleuropneumonia-like organisms, very tiny bacteria that cause some animal diseases. Size and speed of reproduction. Prokaryotic cells are very small, typically only 0.1 to 5 micrometers in size, so small you need a microscope to see them. Because they are simple, they reproduce extremely fast, often doubling in number in 20 minutes under the right conditions. Shapes of bacteria. Bacteria, prokaryotes, come in 4 main shapes, each helping them survive in different environments. Bacillus, rod-like straight sticks, e.g. E. coli. Coccus, spherical, round like a ball, e.g. Streptococcus. Vibrio, comma-shaped, curved, e.g. Vibrio cholerae. Spirillum, spiral, corkscrew-shaped, e.g. Spirillum minus. Structure, cell wall and cell membrane. Most prokaryotic cells have 2 layers outside. Cell wall, a tough protective outer shell giving shape and strength. Cell membrane, a thin layer inside the wall controlling what goes in and out. Exception, mycoplasma bacteria do not have a cell wall, making them soft and flexible. They can change shape easily.
Cytoplasm, the jelly inside.
The inside of prokaryotic cells is filled with a semi-fluid substance called cytoplasm. This is where all the important chemical reactions happen that keep the cell alive. Cytoplasm contains water, nutrients, enzymes, and other molecules. Genetic material. Prokaryotes do not have a nuclear membrane. There's no nucleus bag around their DNA. Their genetic material is naked DNA, meaning it's free in the cytoplasm. They usually have a single circular chromosome which contains all the instructions for running the cell. Plasmids, extra DNA with special trait. Many bacteria have plasmids, which are small circular pieces of DNA separate from the main chromosome. Plasmids give bacteria special abilities. For example, they can carry genes for antibiotic resistance, making the bacteria survive medicines that would otherwise kill them. Scientists use plasmids in genetic engineering to insert new genes into bacteria, a process called bacterial transformation. No membrane-bound organelles. Prokaryotic cells lack complex membrane-bound organelles, e.g. mitochondria, Golgi bodies, lysosomes, found in eukaryotic cells. The only organelles they have are ribosomes, which are small and make proteins but do not have membranes. Inclusions, storage spots. Inclusions are small storage spots inside prokaryotic cells. These inclusions can store nutrients, waste, or other useful substances for when the cell needs them. Mesosomes, special folds of the cell membrane. Mesosomes are unique to prokaryotes. They are infoldings of the cell membrane into the cytoplasm. Mesosomes help with cell division, separating DNA into new cells, DNA replication, copying the DNA, respiration, energy production, acting like a simple version of mitochondria. Think of mesosomes like pockets or folds inside a bag, giving more surface area to get important work done. Complete detailed summary of prokaryotic cells. Prokaryotic cells are the simplest and most ancient type of cells. They lack a true nucleus and membrane-bound organelles. Examples include bacteria, blue-green algae, mycoplasma, and PPLOs. They are tiny, usually 0.1 to 5 millimeters, and can multiply very fast. Bacteria come in 4 main shapes: rod, bacillus, sphere, coccus, comma, vibrio, and spiral, spirillum. Most prokaryotes have a cell wall and cell membrane, except mycoplasma, which has no cell wall. The inside of the cell is filled with cytoplasm, the jelly-like fluid where life processes happen. Their DNA is circular and free-floating in the cytoplasm nucleus. Many bacteria have balls of DNA called plasmids, which carry useful genes like antibiotic resistance. No complex organelles exist in prokaryotes except for ribosomes to make proteins. They have inclusions for storing important substances. Mesosomes, special folds of the cell membrane, help with important tasks like energy production and cell division. This summary covers every important detail in a simple, clear, and complete way, so you understand exactly what prokaryotic cells are and how they work. Would you like a comic strip, flashcards, or practice quiz on this? Of course, here's a great, deepest, most detailed yet simple summary of everything about the cell envelope and its modifications in bacteria. Every key point so you fully understand it. What is the cell envelope? In most bacteria, the cell envelope is a complex, multi-layered protective covering wrapped around the cell. It acts like a three-layered shield, giving bacteria shape, protection, and control over what enters or leaves the cell. The three main layers of the cell envelope are 1. Glycocalyx, outermost layer. 2. Cell wall, middle layer. 3. Plasma membrane, innermost layer. 1. Glycocalyx, the sugar coat. The glycocalyx is the outermost layer made mainly of sugars, polysaccharides, sometimes mixed with proteins. Its composition, what it's made of, and thickness vary between bacteria. Thin and loose, called a slime layer. Thick and tough, called a capsule. Functions of glycocalyx: helps bacteria stick to surfaces like teeth, forming dental plaque or tissues in the body. Provides protection from drying out, desiccation. Shields bacteria from being attacked by the immune system. Can help bacteria escape immune cells trying to destroy them. 2. Cell wall, the strong frame. Beneath the glycocalyx is the cell wall, a strong, rigid layer that determines the shape of the bacterium, rod, sphere, spiral, gives structural support like the frame of a building, prevents bursting if water enters the cell by withstanding high internal pressure, prevents collapsing if water leaves the cell. The main component of bacterial cell walls is peptidoglycan, a tough mesh of sugar and protein molecules. The structure of the cell wall is different in two groups of bacteria. Gram-positive bacteria, thick layer of peptidoglycan. Gram-negative bacteria, thin peptidoglycan layer plus extra outer membrane. 3. Plasma membrane, the selective barrier. The plasma membrane is the innermost layer lying just inside the cell wall. It is made of a phospholipid bilayer with proteins embedded in it. The plasma membrane is selectively permeable, meaning it controls what enters or leaves the bacterial cell, only allows specific molecules in or out, like nutrients and waste products. The plasma membrane also helps the cell sense and respond to signals from the environment. Structurally, it is similar to the plasma membrane in eukaryotic cells, like human or plant cells, which shows a shared basic plan among all living things. The cell envelope as a single unit. Though each layer has its own distinct job, they all work together as one protective unit to keep the bacterium safe, stable, and functioning properly. Without this envelope, bacteria would not be able to maintain their shape, protect themselves in harsh environments, control what goes in and out of their cells. Gram stain and classification of bacteria. The differences in the cell envelope are used to classify bacteria with the Gram staining test, developed by Hans Christian Gram. Uses a purple dye to test bacteria. Gram-positive bacteria have thick peptidoglycan, stain purple. Gram-negative bacteria have thin peptidoglycan plus outer membrane, do not hold the purple stain well, appear pink-red after counterstain. This classification is important for medicine because Gram-positive and Gram-negative bacteria often respond differently to antibiotics. Gram-negative bacteria have an outer membrane that makes them more resistant to some drugs. Complete, deep, simple summary. The cell envelope of bacteria is a three-layered protective system. 1. Glycocalyx, sugary outer coat, can be a thin slime or thick capsule, helps in sticking to surfaces and protecting against the immune system. 2. Cell wall, rigid, gives shape, strength, and protection from bursting or shrinking, made of peptidoglycan. Plasma membrane, inner layer, a selective barrier that controls what comes in or goes out of the cell. All layers together protect the bacterium like a suit of armor, allowing it to survive in many environments. Differences in cell wall structure help classify bacteria into gram-positive or gram-negative, which affects how they cause disease and how we treat them with antibiotics. The plasma membrane of bacteria is similar in structure to membranes of more complex organisms, showing unity in the building blocks of life. Would you like a comic strip, diagram, or interactive quiz on this to reinforce your understanding? Of course. Here's a great, deepest, detailed, yet simple summary of everything about mesosomes, chromatophores, flagella, pili, and fimbriae in bacteria, all in one place, so you understand it fully. Mesosomes, the folded machines inside bacteria. What they are, mesosomes are folds of the plasma membrane inside bacteria, like little pockets or waves. What they can look like vesicles, tiny bubbles, tubules, tiny tubes, lamellae, thin sheets. What they do, build the cell wall. Mesosomes help make and repair the rigid outer structure. DNA work. They help copy DNA before cell division and distribute DNA to new cells. Energy production. Mesosomes help bacteria breathe, respiration, by making ATP energy since bacteria lack mitochondria. Secretion. Help move substances in and out of the cell. Increased surface area. More membrane folds mean more area for important chemical reactions. And provide more space for enzymes, proteins that speed up reactions. Key idea. Mesosomes are like tiny power stations, construction sites, and delivery centers inside bacteria. Chromatophores, solar panels inside photosynthetic bacteria, found in cyanobacteria, blue-green bacteria. What they are? Special membrane folds in the cytoplasm called chromatophores. What they do, contain pigments like chlorophyll that capture sunlight, help bacteria make food from sunlight, photosynthesis, just like plants. Key idea, chromatophores are bacteria's mini solar panels turning sunlight into food. Motility, moving around with flagella. Some bacteria can move, they are called motile bacteria. Others cannot move, non-motile bacteria. Motile bacteria have flagella. Long, whip-like tails sticking out of the cell wall. They spin like tiny motors, pushing the bacteria through liquids. Parts of a flagellum. 1. Filament, the long, tail-like part waving outside the cell. 2. Hook, connects the filament to the motor. 3. Basal body, the motor inside the cell wall and plasma membrane that spins the flagellum. Number and arrangement of flagella vary. Some bacteria have one flagellum, some have a bunch at one end, some have many all around. Key idea, flagella are like motors on a boat. Pili and fimbriae, sticking and connecting, not moving. Bacteria also have two other types of surface structures. 1. Pili, longer, tube-like structures. 2. Fimbriae, short, stiff, hair-like bristles. Pili, made of a special protein called pilin, used for sticking to surfaces or for transferring DNA during a process called conjugation, bacteria sex, like ropes or straws. Fimbriae, shorter and thinner than pili, more numerous, many per cell, used mainly for sticking to surfaces like rocks in streams or host tissues, like tiny Velcro hairs. Important difference from flagella. Flagella for movement, motility. Pili and fimbriae for attachment, not for movement. Complete, deeper, simple summary. Mesosomes are folded parts of the plasma membrane inside bacteria. They help build the cell wall, copy and distribute DNA, make energy, respiration, secrete substances, increase membrane area for reactions. Chromatophores are special folds with pigments in cyanobacteria to capture sunlight and make food. Motile bacteria use flagella, long tails with filament, hook, basal body, to swim. Pili are long tubes that help bacteria stick to surfaces or exchange DNA. Fimbriae are short, bristle-like hairs that help bacteria attach to things but do not help them move. Together, these structures help bacteria survive, move, stick, reproduce and thrive in many environments, from rocks in rivers to inside human bodies. Would you like a comic strip, diagram or quiz on this to make it even clearer? Of course. Here's your great, deepest, most detailed summary in simple, clear words. Like a perfect recap of everything you need to know about ribosomes and inclusion bodies in prokaryotic cells, bacteria. Ribosomes, the protein factories of bacteria. What ribosomes are? Ribosomes are tiny molecular machines inside bacterial cells. Their main job is to make proteins, which are essential for building the cell and carrying out life functions. Where they are found? In prokaryotes, bacteria, ribosomes are mostly found floating in the cytoplasm. Some ribosomes attach to the inner surface of the plasma membrane. Size and structure. Ribosomes are very small, about 15 nanometers wide by 20 long. You need a powerful microscope to see them. Each ribosome is made of two parts, subunits, a small subunit, 30S, a large subunit, 50S. When these two join together, they form a complete 70S ribosome, which is the type found in prokaryotes. Note the 30S, 50S, and 70S states, which are how fast particles are spinning, centrifugation, not simple addition. Function of ribosomes. Ribosomes read the instructions carried by mRNA, RNA, like a recipe. They are amino acids in the right order to make proteins. Proteins made by ribosomes do everything from building the cell wall to carrying out chemical reactions. Polysomes. Sometimes many ribosomes attach to a single mRNA molecule at the same time. This chain of ribosomes working together is called a polysome or polyribosome. This setup speeds up protein production, allowing the cell to make many copies of a protein quickly. Key point: ribosomes make proteins, polysomes make lots of proteins fast. Inclusion bodies, the storage bins of bacteria. What inclusion bodies are? Inclusion bodies are storage spots inside bacteria where the cell keeps extra materials for future use. They float freely in the cytoplasm and are not surrounded by membranes, unlike many storage structures in animal cells. What they store? Different inclusion bodies store different types of reserve materials. Phosphate granules store phosphates, which are needed to make important molecules like DNA and ATP, energy. Glycogen granules store glycogen, a form of sugar that the cell can break down later for energy. Cyanophycean granules, found in cyanobacteria, blue-green bacteria, to store other nutrients. Why inclusion bodies matter? They act like pantries or storerooms for the cell, keeping important supplies ready for times when food or nutrients outside the cell run low. Gas vacuoles, floating devices in certain bacteria. What gas vacuoles are? Some bacteria, especially photosynthetic bacteria like cyanobacteria, purple or green sulfur bacteria, have gas vacuoles inside them. What they do? Gas vacuoles are tiny air-filled sacs that let the bacteria float up or sink down in water. This helps bacteria find the right amount of sunlight or nutrients by moving to the best depth in the water. Example: gas vacuoles work like mini life jackets or inflatable tubes, helping bacteria stay at the perfect level in water. Complete, deep, simple summary. Ribosomes are small machines made of two parts, 30S plus 70S, that read mRNA instructions and build proteins from amino acids. Polysomes are chains of ribosomes working together on the same mRNA, allowing many proteins to be made quickly at the same time. Inclusion bodies are floating storage bins in the bacterial cytoplasm, storing reserves like phosphate, sugar, glycogen, and other nutrients. They have no membranes. Gas vacuoles are small gas-filled structures in some photosynthetic bacteria, letting them move up or down in water to find the best light or nutrients. Overall key idea: ribosomes and polysomes make proteins. Inclusion bodies store essential materials. Gas vacuoles help bacteria move vertically in water. All these help bacteria live, grow, and adapt to their environment. Would you like a comic strip, diagram, or quiz on ribosome inclusion bodies to test your understanding?